Our guest speaker today is me. And so thanks for coming. And I think we're having some minor technical difficulties, so I've been asked to stand behind the podium. So forgive me when I seem so far away. It's not my intention to make it seem that way. But I have a simple goal over the next 30 to 40 minutes, and that's to change your mind and your thinking about thrombosis. I think it's important to do that, even though it's evolved very, very nicely over the past several millennia. I think we're starting to learn some things now that will be really the background or the backdrop for clinical trial for you to comment as well. I don't have any conflicts of interest for this particular discussion, but this is what I'd like to challenge with you. And this is something that has been said to me before and perhaps has been said to you previously. And it's a tenet, I think one that's in evolution, and that is an effective antithrombotic drug by definition and by design will and must be associated with bleeding complications. And I think if we look at thrombosis and coagulation in a traditional sense, absolutely. If you're developing a drug and you don't see a bleeding signal in development, you wonder if that's really going to have an effect in clinical trials. There have been lots of drugs that have been killed in phase 2 because there was not a signal of bleeding, particularly with accelerating doses. But I think if we start to look at thrombosis differently, that this tenet will no longer hold true. And the way we design clinical trials will have to change as a result. It makes it a little bit more complex. So let's start with, again, one of the traditional paradigms of thrombosis, the Vercaus Triad. Those of you probably are familiar with this. You've heard it over and over again, perhaps from me, perhaps from others. Rudolf Virchow was a pathologist, a clinician, did a variety of things, including describing the first leukemic cell, was the first to suggest that cells were born of other cells, so it was a cell-based model of disease. And then a little bit later, he started to do some work in thrombosis, and it was the first to connect the dots in terms of deep vein thrombosis and pulmonary emboli, called them embolia. So he coined that term for the first time. But in addition, he started to set the stage for what the environment or the conditions would need to be for thrombosis, and basically said that you required 3 different complementary components. That is, an abnormality within the blood, something at the level of the vessel wall, and an abnormality in blood flow. And this paradigm fits in many, many diseases and conditions that are characterized and typified by having a thrombotic type of phenotype. Malignancy, trauma, pregnancy, childbirth, acute coronary syndrome, it all fits. Now this may be difficult to see from the back because it's difficult for me to see from the front. But there's historic value here, and that's why I've included it. And this is the description of the Waterfall Cascade, and was actually the end result of decades of basic biochemistry and protein-protein chemistry by a number of individuals. But this was described concomitantly by 2 groups. It was Davies and Ratanoff. They were working at Western Reserve. That's before it was Case Western. And then at the same time by McFarland, who was at Oxford. And there is a Duke connection here. As it turns out that Davies had been working with an investigator by the name of Hans Neurath, who is a protein chemist here at Duke. And this goes back into the 1930s and the 1940s. So there is a very close connection between Duke, not unexpected, and understanding coagulation and thrombosis. Now this waterfall sequence of blood clotting, as many of you know, it starts at the top, and this is a series of biochemical steps with autoactivation where you have inactive zymogens being converted to active proteases. And the intrinsic cascade, as first described, and this was simultaneously published in Nature and in Science in 1964, another very interesting story in that Ratanoff and Davies had submitted to Nature and the paper was rejected. They turned around, didn't make any revisions, they sent it to Science. And when it was published in Science, McFarland's same waterfall cascade was published in Nature. They turned out to be simultaneous publications. But you start with factor 12 to 12A, and then plasma thromboplastin accelerator, which was later named factor 11, then factor 9, and then factor 10, Stewart factor. And if you ask who's Stewart, another bit of North Carolina history. He was a person from North Carolina who was seen at UNC because of a bleeding disorder. And the genetics is interesting. It turns out that his parents were cousins. So there is a inheritance there that defined a factor 10 deficiency, the Stewart factor deficiency. And ultimately you go down the waterfall cascade. Now in the bottom panel, the following year, Davies, and this is a review that actually was published in 2003, but it's 1965, where he starts to describe the extrinsic cascade of tissue factor. So you had something that is external or extrinsic to flowing blood. It was tissue factor found within vessel walls that were damaged. On the intrinsic side, something that was intrinsic to the blood was responsible for coagulation. But in terms of these converging or non-converging pathways, that's where the paradigms start to change, and some of the targets for the development of anticoagulants that may not follow that tenet of bleeding as a prerequisite for drug development come into play. So let me talk with you about that a little bit. This is the way we currently look at the coagulation cascade on cell-based surfaces. And this is work done by Maureen Hoffman across the street at the VA. The original work was done when Maureen was at UNC with Mac Monroe and Harold Roberts. Harold Roberts being one of the true contemporary fathers of coagulation, along with a number of people at UNC. And it takes the biochemistry of coagulation and puts it on cell surfaces. And you can see in the upper left-hand corner that you have a tissue factor-bearing cell. Traditionally, this would be something that one would find within a vessel wall. So again, extrinsic to blood itself, but it could be transformed endothelial cells or monocytes or macrophages. You have tissue factor and factor 7A. You have conversion of 10 to 10A. You have a small amount of thrombin that is generated that activates the platelet and amplifies the entire process. And then ultimately on the surface of the activated platelet, you have further assembly with factor 9 and factor 8 in complex with one another, and you have this burst of thrombin generation. But you'll notice in this cell-based model that you don't see factor 12. And you say, well, that probably is because it really is not a cell-based phenomenon. So how does that actually fit? And that's where the story becomes particularly interesting for drug development and things that are going on in Bruce Sullinger's laboratory right now. So in that context, I've shown this slide before to the fellows and in presentations previously, and I believe that it helps us start to change the paradigm a little bit in terms of thrombosis. Not to say that we're going to take the waterfall cascade and all the biochemistry out of it, but we're looking at other mediators that one can leverage as potential targets in the development of new drugs. And some of the targets include polyphosphates that are released from endothelial cells, white blood cells, and platelets. It's something that stimulates coagulation, microvesicles from a wide variety of sources. Spent a lot of time talking about platelets. And I've also presented to this group before some of the thoughts around nets, neutrophil extracellular traps, as something that might be a unifying theme around thrombosis in acute coronary syndrome, in pregnancy, after delivery, trauma, and malignancy. And I'll develop those themes a little bit more with you. But here's the point that I believe is important in terms of taking millions of years of evolutionary biology and some of the things that we've learned about the biochemistry and starting to tease things out. And this paper that was in Nature in 2010 does that very nicely. It starts at the top with polyphosphate, RNA, or foreign surfaces. So this is part of your contact system that isn't glass or beads or silica or any of the things that can be used inside a laboratory. This is basic biology of where contact activation becomes important. And this is the piece around sepsis, around mechanical heart valves, around guide catheters sitting in the left main coronary artery, all of your contact activators. But take notice of this. As we start to move down the cascade a little bit farther, triggers of thrombosis but not required for hemostasis. Now we have this uncoupling, and this is where the paradigm starts to change, and that tenet that in order to have an effective antithrombotic agent that it has to cause bleeding. And you can see where this is going in terms of if you choose the right targets, if you broaden the paradigms of thrombosis, you get yourself out of that quandary of everything that is effective having to cause a certain amount of bleeding. But in addition to that, it shows factor 12 and factor 11, and it's very, very important to interface with inflammation. And this starts to broaden the scope. Inflammation and thrombosis in acute coronary syndrome. How many times have you heard that? You don't have to go far to connect those dots. Inflammation and thrombosis in somebody with septic shock or following major trauma. These are traditional paradigms that are playing out essentially every day. As you go farther down and you're looking at tissue factor and the common coagulation pathway factor 10 as an example, that's where you have a requirement for both hemostasis and thrombosis. So you would say, looking at this representation, that if one were to choose these targets, then perhaps you could uncouple bleeding and thrombosis and the development of drugs that do not require a bleeding signal to show that they're effective. I'm not going to spend a lot of time to say that there is an evolution of coagulation where there is gene duplication, there are gene dropouts, there's a lot of redundancy, but as you follow vertebrate evolution, you'll see that there is a series of consistent or homologous proteins that are present as you get farther and farther along the evolutionary path. But at the same time, when you look at contact activation, it's much more recent. And I've mentioned this to many of you previously. The evolution of coagulation goes back about 350 million years, but the contact activation pathway is recent. It's about 150 million years ago. And one has to ask, was there a particular reason for that, or was it just by chance? And I guess either one is possible, but in terms of the link between inflammation and thrombosis, perhaps over time that there was a greater need to have contact activation. There was a greater need to have more immune responsiveness because people or our predecessors were being exposed to things. But yet when you look at this whale off the coast of Cape Cod, they don't have factor 12. nor do dolphins. So it's not required for mammals. I presented this and said, you know, could we work together to understand the evolutionary biology? And I said, well, it's fairly simple that whales don't run into stuff, so they don't need contact activation. But I think they could run into stuff, or people could run into them, so they might need it, but it's not required. But one thing that is clear, that in terms of evolutionary biology, there are a lot of similarities between coagulation factors, factors that are required for the breakdown of clots, the fibrinolytic pathway, and you can see that in the protein homology. There's lots of diversity around contact activation, and this is important, and this is information that goes into the work that the Sullinger Laboratory is doing right now. Becky Woodruff, who's one of the graduate students, and we're going to have her come and go into a little bit more detail of the work that she's doing. But to suffice it to say that when you look at factor 12, and we look at some of the potential stimulators, that polyphosphates that could be present in activated platelets or other cell types, collagen, or negative surfaces that you might see for cell-free DNA, as an example, in sepsis and trauma, that there's activation of factor 12. And then you have this very, very complicated interplay of fibrinolytic compounds, such as urokinase plasminogen activator, tissue plasminogen activator, bradykinin, all of the inflammatory components and innate immunity components that we're familiar with. And then on the other side, argue that this factor 11 is a better target because it doesn't have as much interplay with the fibrinolytic system, but it also takes you out of the inflammatory piece that might be important in a lot of disease states. So factor 12 and factor 11 are certainly laudable targets for drug development, and I'll walk you through that in a moment. Looking further at the diversity around contact activation, you can see the importance of interplay. If you're going to factor 11, you have to be aware of some of the other biologic effects that might be seen. As an example, bradykinin, which is important for NO production on the endothelial surface. It's important for vascular permeability as a stimulus for prostacyclin. Then you have that fibrinolytic side of the cascade to be concerned about as well. And there are ways to take advantage of this. One could design an aptomer, as an example, that binds to factor 12 in such a way that it has anti-inflammatory effects, as an example, or solely prevents thrombosis. But again, the information that we have in humans with factor 12 deficiency, they don't seem to bleed. With factor 11 deficiency, they can bleed if you challenge them, but you have to challenge them quite a bit in surgery or major trauma before you see bleeding. But they're certainly not protected against pathologic thrombosis like acute myocardial infarction or stroke. Now, there are a number of factor 11 inhibitors under development. And this is a paper in the Journal of Thrombosis and Thrombolysis just last year that looks at one particular drug being developed by Bristol-Myers Squibb as a factor 11 antagonist. And as Vicki knows, we've been working very, very closely with BMS and their program. And it's an opportunity for the technology that's been developed by Bruce Sullinger with aptomers to be important in modeling. And this is an example of looking at a thrombosis model with increasing concentration of their factor 11 inhibitor in a rabbit AV shunt model, as well as in a venous thrombosis model. So there are factor 11 inhibitors under development, and the attraction is the possibility that you would have that uncoupling of thrombosis and bleeding and changing that tenet that one has to have a bleeding signal in order to have an effective antithrombotic drug. Okay. So obviously we're interested, I mentioned Becky's work with targeting factor 12 and factor 11. One could also target kallikrein as an example. And just as a reminder in terms of the paradigm around aptomers, they're modified single-strand RNA or DNA that target specific sites. This is protein-protein binding sites. This isn't a catalytic site like you might have with your factor 10 inhibitors, rivaroxaban, apixaban, edoxaban as an example. And they have the specificity of a monoclonal antibody. But in addition to that, because one knows the sequence, that one could develop an antidote. And you'd say, well, look, if what you've been telling us is true, that you choose the target appropriately, you don't have to worry about having an antidote. And that may be true, or it might be, as an example, you have someone that you've used a factor 11 aptomer who's going to the operating room, and they're really going to be challenged in a big way. Under those circumstances, you may want to have an antidote, and by Watson-Crick base pairing, which means that you have very, very tight binding, that you dissociate and your effect dissipates. And this complex is eventually cleared by endonucleases. So it is a construct that could be applied to new targets. And just one slide of the work that Becky is doing with a factor 12 aptomer, where she's been able to develop a series of aptomers that target factor 12. We don't know exactly where it binds. Factor 12 is a very, very complicated molecule, despite the fact that it's been around for a while. There's not a crystallographic structure that one could turn to. That might be an opportunity for our U54 interface with centers like Washington University and St. Louis, where they could help us perhaps as protein chemists to identify what the factor 12 sequences are, and perhaps do a crystallographic representation bound to an aptomer to stabilize it and make it easier to do the crystallographic work. So Becky is well on her way to developing a series of aptomers against factor 12 and factor 12A. Now, I mentioned polyphosphate as being something that also seems to be a good target, and one where you might be able to change the paradigm. As best we can tell, polyphosphate, although found in a variety of different cells and a variety of conditions, is not required for hemostasis. You might be able to selectively inhibit thrombosis without worrying about the bleeding part of that. This is a representation from a paper in 2004 looking at localization of polyphosphates in small organelles so that you can induce their presence, and that helps you in designing experiments. This more recent paper in 2012 tells you that you're able to distinguish polyphosphates that come from different cell types. So polyphosphates that originate from red cells versus monocytes versus platelets. And as this study shows, when you look at polyphosphates that come from platelets and also from erythrocytes, that you can show that one generates thrombin as a result of polyphosphate stimulation. Now that's important in terms of being able to have models that you can exploit to do experiments in thrombosis, particularly if you have ways to inhibit polyphosphates. I mean, it's a lot of fun to talk about the biology. It's a lot of fun to talk about new paradigms, but typically we will ask at the DCRI, okay, what are you going to do about it? Do you have a way to inhibit it? Do you have an aptomer? Well, in this case, I'm showing you some of the work that's been done in Bruce Sullinger's laboratory with cationic polymers that bind onto inorganic polyphosphates, and being able to exploit those relationships, which is basically a charge density based relationship, as a potential therapeutic. And let me show you some examples, and this is just one. Shashank Jain was a postdoc in Bruce Sullinger's laboratory, and he performed a series of experiments that were published in PNAS just last year, where they took some PAMAM dendrimers. So these are, in essence, your polymers. They're polyamido amines, these that have been used for years in drug delivery systems, but one could also use them for direct therapeutic effects. So this is in a model. There are a couple of models that were utilized here. One was a collagen-based thrombosis model, and the other was a collagen epinephrine-based pulmonary embolism model. And on the left-hand side, what I'm showing are the number of animals that survive in the presence or absence of these PAM-AM dendrimers, and then a representation of the antithrombotic effect. So basically what Shawshank was able to show is that in the presence of the PAM-AM dendrimers, or if it's easier just to look at them as polymers, that despite that very substantive stimulus for thrombosis, that you could prevent thrombus formation, that you could prevent the death of the animal. This is in a small animal model. And then on top of that, in the lower panel, you can look at whether bleeding was caused or not. And as you can see, with the PAM-AM, this is G3, that just means the third generation. You go from 0 to, now we're up to 11, these very, very large molecules that could be used for a variety of things, not for antithrombotics. But that's what the G stands for. So you have it both ways. You have a reduction in the likelihood of thrombosis in a very powerful, provocative model. You have a mortality reduction, if that helps for a DCRI related conference. You have a reduction in mortality of these mice. In addition to that, you've uncoupled thrombosis and bleeding because you've attacked thrombosis in a different way. You've attacked part of it that is not involved in hemostasis. So the tenet of requiring bleeding as a signal for an antithrombotic starts to fall apart. And when you broaden your paradigms of thrombosis, and this is just one example. This is something that I've shown previously, and Matt Sherwood has been working on this and has several different experiments that will be performed with the help of Bruce Sullinger. And it makes, or it represents another example of looking at thrombosis in a different way, to change your mind about thrombosis. Don't forget about the biochemistry, don't forget about factor 10 and thrombin, but start to see it differently. And in this particular case, it's cell-free nucleic acids, where certainly it's been shown in the oncology sector that you have cell-free DNA in very large concentrations, and you can interrogate them to look for mutations, epigenetic studies of methylation, microsatellite changes. Same thing in myocardial infarction. There is cell-free DNA in this particular patient population and also in individuals with stroke. So this could be a diagnostic, or it could be a target for the development of a new generation of antithrombotics. This is a classic paper by Fuchs and others looking at extracellular DNA traps in thrombosis. The paradigm here is that you stimulate a white blood cell. You can stimulate it in a variety of ways. You can use lipopolysaccharide to simulate sepsis or PMA, or you can use things like peroxides and other types of materials that one could find in humans under varying conditions. And when you look closely, after the neutrophils are stimulated, you have these long strands, and you have platelets and red blood cells that are adherent to the strands. The strands are consisting of chromatin that have been released from the white blood cell, basically unravel and unfold. And you have the adherence of red cells and platelets, and as you can see in the lower hand corner, that in the presence of DNA ACE, that essentially the whole thing falls apart. So it is DNA strands. It's part of the nuclear materials that have been released. And this is very important teleologically as a defense mechanism for containing infections. But also, when you look at what role it might play in thrombosis, again, this is Fuchs and others, this is in a primate mechanical injury model of deep vein thrombosis. And you can see in this particular case, here's the thrombus in the iliac vein. And when you stain it for DNA, this is called gogoblue staining, and this is histones, that actually the thrombus and the adherent portion of the thrombus to the vessel wall are traps, that it's DNA and histones on these long strands. You could say that the same is true for von Willebrand protein, which is in the center. Another opportunity for collaborations in our U54 group as we look at von Willebrand factor and ways to inhibit it. But the story is important at so many levels where we're focusing on thrombosis. I'll just show you one recent study that's published in JBC, the Journal of Biological Chemistry, within the last year to make some important points about, does this really play out in humans? And the answer is, it does. These are thrombi from 3 different patients. One is a person that has a popliteal thrombus that is removed by the vascular surgeons with a catheter. The second is derived from a person with an abdominal aortic aneurysm where there's mural thrombus in a majority of those. And the third is in a person that has a fem-pop bypass thrombosis where it's surgically removed. In each particular case, the first stain is fibrin. You say, okay, oh, old news. And I agree. The second is a histone stain. And you can see in humans where the thrombus has been removed that you have histones that are present. There is DNA present. When you combine them with both of the stains, that they are in complex. And when you look with electron microscopy, you see nets, you see fibrin, you see histones. They're all present in human thrombosis. So these are not just pie-in-the-sky suggestions as new paradigms. These are playing out, and the literature is really starting to accelerate, and we need to be part of that, and we are part of that at Duke. So this is a great example. If you have an idea, don't wait, because somebody elsewhere has a good idea, and you say, well, that's fine. If somebody else finds that there happen to be cell-free DNA in human coronary thrombus, that's okay. It would have been nice if we did it first. Is that right, Matt? When we were discussing this 2 years ago? I'll take full responsibility for that. So some of our colleagues in the Netherlands published just this year in Thrombosis and Hemostasis. If you do extraction thrombectomy in patients with acute ST segment elevation myocardial infarction, and you use a gogoblue stain, that you see DNA, you see nets, you see histones, it's all present. And targets for intervention, not just the biology, but targets. Then one last study. This is in an animal model of acute lung injury to once again show that when you do staining, and there are lots of different models of acute lung injury, but in this particular case, they're looking at a very important component of the inflammatory thrombosis paradigm. And these are danger-associated molecular pattern recognizes particular shapes of things. So it's not just receptor-mediated, but there's certain shapes that the body recognizes as being potentially dangerous. And this is a group of those proteins known as high mobility group box, or HMGB1. And you can show that actually when you stimulate a white blood cell with this particular substance, that you produce nets. And these are not subtle. These are actually quite profound. It doesn't take electron microscopy to see them. So this is with a gogoblue stain, the DNA, the histones, and then both of them in complex. And you can also show that with lipopolysaccharides, so this is your sepsis model, and then also with PMA as a stimulus. But the greatest stimulus for net formation seems to be with this HMGB1. So another potential target and example, in this case of an acute lung injury model, that these are players. I showed you some of the work that has been done in the Sullinger laboratory in terms of polymers binding onto polyphosphates. Polymers also can bind onto cell-free DNA, cell-free RNA, and varying complexes. So the story starts to take shape that there are experimental observations. Now there are human observations, and you could say that they're validation that this is something that plays out in human conditions and disease, and it does, and also the possibility that there would be targeted therapies, again, that would leave hemostasis alone. As best we can tell, these cell-free DNA negatively charged surfaces are not required for hemostasis, but they very much play into thrombosis in trauma, in sepsis, in acute coronary syndrome, in pregnancy and the complications of pregnancy, and in malignancy. And one might say this is the common thread for each one of those conditions as to how do you get from Vercaus Triad to thrombosis? Well, you just change around some of the components, but they're still there, but they have new names. And those new names are DNA, histones, and those in complex. So I'm going to leave you with a thought and a change in paradigm. And that is an effective antithrombotic drug, by definition and by design, will not and fundamentally must not increase the risk of bleeding. Now, you might say, we've heard this before, trying to uncouple thrombosis and the likelihood of bleeding, but hopefully I've changed your mind just a little bit about thrombosis, and I've changed your mind a little bit about targets, and I've certainly hoped that I've changed your mind that there isn't an opportunity for antithrombotics. I've heard that we're done. We've been there, we've done that. The factor 10 inhibitors, there's nowhere else to go. We have a lot of work to do, and it's just begun. So if you want to get on the train is getting ready to move. And if translational science actually means what it's supposed to mean, and that is you can take some fundamental constructs and you can translate them to patient care and advancing and optimizing how we take care of our patients, this is the train that I believe that you would want to be on. So thanks for your attention, and I welcome any thoughts or questions that you might have. Good. Hopefully now I can come out from behind that big thing. So.
Thanks, Dr. Becker. I had two quick questions. First of all, how are the genetic elements responsible for thrombosis and thrombolysis related in the genome? And how have they evolved so that there's such an exquisite balance between the two? Are there any organisms where the balance is a little off in evolution? And second of all, we've known that beta hemolytic strep can break blood clots since the 1920s. Why did it take 60 years for that to be used as a therapy?
Well, let me start with your second point first, because I'm still thinking about your first question, and as I think, I'm going to answer your second. The Staphyloc kinase, streptokinase story is one that's very, very interesting. And if you think fundamentally, so make believe that you're a staph or a strep right now. And you're part of an infection. The last thing that you want to do is be contained. So what does the kinase part of that actually do? It breaks down nets. It can dissolve them, and infections can disseminate. So we can connect those dots pretty easily. In terms of the genetics behind these constructs, again, from my perspective, if we just look at contact activation, I would have to think that in part that it's evolutionary in terms of having greater immune capabilities. But in addition, it could be in part containing infections because it does that very, very well. There's a a link between contact activation, polyphosphates, and net formation, and one can show that fairly easily, and several groups have shown that. But factor 12 is not required. It's not a lethal mutation. That's an example of evolutionary biology where you have gene dropout that's not a lethal mutation by any means, and certain species like whales and dolphins seem to do just fine without it. So why humans seem to need it as a little bit more may be the kind of exposures that we have. But it is an area of investigation, and it'll take people much smarter than I am to understand the evolutionary biology of that and where it actually started to branch out. And you started to see some species, mammalian species, with and without. It's a good question. A smart person like you could be thinking about that, Tarek. Yeah. So what do you think, Chris? Is this...
So it's terrific. Congratulations. It's really, and you put it together so nicely. It's it really is exciting to be provocative in a way that's so well supported to kind of push us to a different level of understanding and investigation. Let me make a couple of observations and see what your response is to these and how they might fit into this. One is we see this really interesting phenomenon of unexpected predictors of bleeding and thrombosis. For example, this high sensitivity troponin level in the blood seems to be a good predictor of bleeding. Now, whether that's just that it's vascular health or independent of other factors, it's and of thrombosis. It's interesting. And then my other comment was around the issue of bleeding and new and old targets for anticoagulation, and how the kind of context and the dose seem to be so critical in the balance, and you know, whether or not this might be different. Maybe this would be different is part of your point, that, for example, you take apixaban 5 milligrams twice a day, and you give it with clopidogrel and aspirin, and you get a 5-fold increased risk of intracranial hemorrhage and a tripling of major bleeding. And you give it for people who've had a DVT and several months later, and you compare it to placebo, and it has less bleeding than placebo, but with very small numbers. But anyway, it seems to be quite safe, depending on the context and the antithrombotic therapy that it's given with.
Your points are critical, and we're early into the study, as you can see. Factor 12 has complexities, and just because of its link with so many things, with bradykinin, with the fibrinolytic systems, and there's up and down regulation. So it may not be the optimal target because of those complexities. Factor 11, a little bit cleaner, as I mentioned. The question, of course, would be how much does it take to get the effects that you want, but not to push to the point that you're getting unwanted effects. And the same thing with the polymers and polyphosphates. We'll learn more about hemostasis. I think it would be naive of me to say that we can completely uncouple them, because they're probably connected at varying places. And if anyone in the audience is a polymer scientist, they'd say, no, there are other things to worry about with polymers. So it's not a free ride by any means, that there are antiplatelet effects that have been shown. Some of them that are lethal. So if you take some of these polymers and you inject them into mice, that they develop lethal pulmonary thrombosis because of platelet activation. So we have to work through all of those. As far as that, similarly, you made the point about inflammation and as a predictor of bleeding. I think there's probably a message there that gets down to vascular integrity and a variety of things that we could probably learn from, whether it's in the context of atrial fibrillation or in others where the inflammatory response is a little bit more profound. And I think that particular part of how the drugs work, the thrombosis inflammation interface is important, something that we have to understand a little bit better on the safety side as well as the efficacy side. So, yeah. Okay.
Things that we can learn in rare diseases and populations of people who have hemophilia or TPP or very rare and genetic and maybe problems that evolve from consanguinity that will inform the therapeutic options that you develop for people with stroke and thromboembolic, chronic disease.
Yes.
And whether, so that's what I'm wondering, and particularly because could we work with places where there is a high prevalence of consanguinity to develop a sort of experimental cohorts of research subjects, not just for our own new knowledge, but actually to develop a greater confidence in ways that we would help those populations improve their population health.
Vicki, your point is critical, and I believe in part that we have taken advantage of understanding human conditions. So whether it's in hemophilia A, which is your factor VIII deficiency, hemophilia B, which is factor IX, which was part of the platform for the factor IX aptamer development, is to say, we know a lot about hemophilia B, and let's try to utilize that as we develop drugs. And hemophilia C is factor XI deficiency. So we have taken full advantage of that. I think that beyond the obvious, that where research is going, and the geneticists, I mean, if Svati Shah were here, she would tell you that we're starting to look at biology and mechanisms by first understanding the genetics, then the complementary changes in metabolomic profiles, and then with recall types of studies where you've defined a little bit about the signature, but you call people back and you do much more deeper dive of sequencing, whole exome, whole genome sequencing, and then in that same context start to look at some interventions. So the same approach could be taken for some of these, for sure, and the informatics piece will be vital to that undertaking. So, okay. Good. Well, I hope you found some of that, or maybe even all of that, interesting, and certainly in the future, it'll be our goal to continue to present some of this translational work and hopefully get to the point where there are interventions that can take full advantage of the incredible strengths and insights of a DCRI at early phase, and then maybe like the factor IX aptamer that's on the verge of starting phase 3, that you'll have one of those true bench to bedside moments that we don't get that often, but we'd like to be part of it. So thanks very much.